A very good afternoon to all the participants present over here. On behalf of the organizing committee, I would like to welcome you all in the 5-day FDP on recent developments in the fields of computational biology. So today we have our speaker as Dr. Shubhadeep Basu. Now I would like to introduce him. Dr. Shubhadeep Basu is a full professor in the Computer Science and Engineering Department of Jadavpur University since 2017. He received his PhD in engineering degree from Jadavpur University in 2006 and joined there in the same year. He did his postdocs from the University of Iowa, USA in 2010 and from the University of Warsaw, Poland in 2012. He also holds an honorary position as a research scientist at the University of Iowa, USA since 2016. Dr. Basu has more than 230 research publications in notable international journals and conference proceedings in the areas of pattern recognition, machine learning, bioinformatics, biomedical image analysis, etc. He has also edited 9 books and has 2 US patents. Dr. Basu has worked in reputed international institutes like Hitachi Central Research Laboratory, Japan, University of Iowa, USA, Bournemouth University, UK, University of Lorraine, France, University of Warsaw, Nensky Institute of Experimental Biology, Poland, and Hanover Medical School, Germany. He has supervised 8 PhD students and received several major research grants from UGC, DST, DBT, Government of India. Dr. Basu is a recipient of the Research Award from UGC, Government of India in 2016, and the DAT Fellowship from Germany in 2018. He has also received a Boys Cast and Fast Track Fellowships from DST, Government of India, HIVIP Fellowship from Hitachi, Japan, EMMA and Plink Fellowships from the European Union. He is the current chairperson of the Computer Chapter, IEEE Kolkata section, a senior member of IEEE and life member of UPRI. So now I am requesting Dr. Shubhadeep Basu to take over the session and please deliver his speech.
Thank you very much for your kind introduction, and very good afternoon to all of you for inviting me for this lecture. Okay, so let me first try to share the screen. Just a minute. Is it visible?
Yes, it's visible, sir. Yes, please continue.
Just a minute. Okay, so very good afternoon, everyone, and today I'm going to deliver a lecture on bioimage informatics. Okay, so bioimage informatics, and especially what are the recent methods and applications on dendritic spine plasticity analysis. So more specifically, I'll discuss what do you mean by dendritic spine plasticity and how we because this is in general, this particular FDP is on computational biology, and I'm sure you are hearing from different perspectives of computational biology. And in this particular lecture, I'm going to talk more about like computational biology with respect to image analysis concept. Okay, so we actively work in this bioimage informatics domain, and I can share with you some of our latest research in this domain, and what do you mean by this interesting area of dendritic spine plastocysts, especially in this era of deep learning and with the renewed interests in neural information processing. So we are all familiar with artificial neurons, and we talk about artificial neural networks, ANN. So here I'm going to talk about on the reverse side of it, that is the real neurons, okay, real biological neurons and how they work. Maybe you can get some insights from that, and we'll see that how we are from computational background, right? So I'm personally, I'm from biology. I'm not from the biology background, but how from the computational perspective we can actually go into a biological problem and can solve it. So that would be my prior focus in this particular lecture. So with this, let us move on to the next slide. Just a minute. Okay. So as I said, like what do you mean by bioimage informatics? Okay, so bioimage informatics is a subfield of bioinformatics and computational biology, and these days there are specific subtracks under computational biology and bioinformatics with this particular terminology, that like bioimage informatics, especially when you have lots of, because there is a huge developments that are taking place in terms of imaging modalities, high-end computational architectures are coming up. So we are getting tons of biological images are getting generated. So how you actually analyze those images and make meaningful insights from those bioimaging data, that is important. Okay, so in general, the field of bioimage informatics focuses on the use of computational techniques to analyze bioimages that I said, and especially in this particular lecture we are focusing on cellular and molecular images. Okay, so cellular images, I'll be working mostly with neuronal cells. So in large scale and high throughput experiments. Okay, so we'll be talking with very large numbers and very high resolution in the micron level and nanoscale level. Okay, so very high resolution microscopic data. So automated microscopes are these days able to collect large volumes of data with minimal intervention. So I'll give you some examples, some data I'll show you that actually that are with in vivo imaging, that are in vivo means that can be generated from the live cells, and there are also some ex vivo and in vitro imaging, that means some are like from the dead cells and all. So those types of data will be dealing here, and I'll discuss briefly on different types of data sets that will be that are popular in this particular domain. So as this has led to data explosion, this requires automatic processing. So this is the first slide. So as you know, like there is always a huge need for automatic, semi-automatic processing. So some of the methods that I'll be discussing here are not completely automatic, they are semi-automatic, because in biology, often you need to work very closely with the biologists. And if you generate something completely automatic, it may not completely serve the purpose of the biology. So there is often a practical requirement of selection based analysis. So they need to work on an interactive mode to trigger some automatic processing. So it depends on the application, and I'll discuss something about that. Now, in general, bioimage informatics can be on any aspect, but as I was briefly mentioning, that major focus is on understanding the brain using neuroimage analysis. So we'll focus on mammalian brain, and not just mammalian brain. We'll focus mostly, like all the images that we are working here are on mouse model experiments. So we'll be working, I'll be showing, means we are extensively working with our biological partners to work on mouse model data. As you know, that if you are familiar with bioinformatics and the conservation of the genetic codes in the mammalian family, you'll be seeing that the conservation of the mammalian brains are extremely high. So the structures of a mouse, the neurons in a mouse model are very similar to that of a human or other higher organisms. So most of the drugs and all these experiments are usually done on a mouse model experiment. Earlier, it was done on, say, on a monkey models, guinea pigs models. Now they want to work with even smaller organisms for better management, etc. So people work with mouse model experiments. So in general, I'll discuss all these things as we proceed. And in general, we have different unknown territories while we try to explore our brain. So the major query here is understanding the brain. How do we understand, and how much we understand our brain? So there are several challenges. So general understanding of human brain functions still remain a big challenge to the scientists, and especially the emotional brain, like how emotions work, how the memory work, like how we learn and forget. So how the flexibility of adult brain gradually degrades with the age to intellectual disability are still largely unknown. And this is one of the last frontiers of human understanding in our biology and medical science, that even if we know how different diseases work, and we can replace so many organs, we can replace the heart, we can replace the liver, we can replace a bone so easily, but there is no way we can replace a brain. So when you have a healthy adult, or apparently healthy adult, and still the brain functions are gradually degrading, you have no real option to replace that. So the neurons are so intricate structures, and when the efficiency of the neurons and the overall brain functions, they decay. So we are at the mercy of nature. So that is the problem. So this is one of the areas where people still have very limited understanding. So very limited understanding. So that's the area in general, like the emotional brain. So we have the thinking and feeling of our activities. We have some inputs through our, say, for example, the eyes. So we have the main region, that is the limbic system that we call, that is the amygdala region, the hippocampus, that is responsible for this emotional brain activity. So as I focus mostly in this limbic system region, you can see that probably you have studied somewhere in your biology somewhere that amygdala region is responsible for memory, decision making, and emotional responses, and this hippocampus region is mainly for this memory, navigation, hypothalamus controls body temperature, hunger. So these regions are interesting, and lot of analysis is done on the neurons at this level, especially the amygdala and hippocampal regions, when you study the memory and decision-making abilities, etc. So, as I said, like it is often, the biologists often call that the understanding the brain is probably the last frontier. We have close to 100 billion neurons in human brain, and they are connected to synaptic junctions, and some electric signals are exchanged. So that much we all know, but that is a very crude understanding, very much top level understanding of the biological neurons. So what actually happens, that is that we'll see in the latter part of our lecture. But if we just see this much, that when we have some exchange of electrical pulses, signals, so there are two ways how we can interface with can we interface with the brain? Can we gather that information of those electrical signals? So there are a lot of experiments in that line also. So one of the major areas where people work with different types of EEG signals, and also you all know about Elon Musk, and Elon Musk is also into it, and Project Neuralink is one of Elon Musk's favorite projects, where they are trying to place a small dime-sized chip onto the implant into a person's head. And if you see the normal EEG signal processing, the sensors, they are mostly non-invasive. But the approach of Elon Musk is that is actually invasive. They are making a big, they're making a small hole inside the skull, and they're actually placing it inside the brain. So that is the game changer that they are trying to say, because the human skull is so protective in nature. It's like a big solid helmet, and it can actually it is actually protecting the inside neurons in a significant way, as well as it is very difficult to accurately map the signals, signal exchanges from outside the skull region. Okay, so what Elon Musk and the Neuralink project is actually proposing, that they are trying to make a small hole inside the skull, and they're trying to implant it inside our brain so that they can catch the signals in a greater way. And they are working on an iOS app, and you can there are so many videos and so many demonstrations. They are working on a pig model at this moment, and a monkey model, and they can say they are playing, they can react to something, and there are a lot of advantages to that. So think about someone who is having paralysis, and just by thinking they can move some cursors onto the screen and do some operations. So if you can capture the brain signals, so that can be huge. So in general, the Neuralink app allows you to control your iOS device, keyboard and mouse directly with the activity of your brain, just by thinking about it. So this is more futuristic, but partially this is happening these days. So you can follow this particular link, and it may be an interesting, you can keep track of their developments. But here in this particular lecture, I'm not going to talk more about this, how we interface with the brain. Rather, I'm more interested, can we image the neurons in the brain and analyze them in 2D, 3D, or in 4D? 4D means in time varying 3D data. So this is of more interest to me, and this bioimage informatics lecture that I'm giving, I'll be discussing more along this line. So nanoscale resolution can be achieved using advanced electron microscopy. A lot of different hybrid microscopy is coming up, and also like multi-photon microscopy, like STADE, etc. And even in confocal laser scanning microscopy, also you can achieve several means nanoscale resolution is possible. I can show you some of the images with significantly high resolution, but my images will be mostly from the confocal and the multiphoton microscopy only, not really from the in this lecture, not from the electron microscopy slide. So some examples of the data that we'll be commonly discussing in this particular lecture. So there are in general 3 types of images that we capture. One is called the in vitro images, so where you have the live cells. Okay, so these are all like these branches are the dendritic segments from a neuron. Okay, so these are all images of neuronal cells. And in the neuronal cells, you have the main cell body or the soma. So that is not important in our case because that's a larger structure. But we are interested about the dendrites. So when we have the dendrites, so we have axons of the other neuron that connects with the dendrites of one neuron. So here, this is basically which part of the image that we want to highlight. So everything that the black region is also packed, but it all depends how you actually, these are all fluorescent microscopy. So you stain a particular cell with certain color. So only the region, this particular region is highlighted. Definitely there are other parts in this black regions which are not highlighted. That is why we see the images like this. Okay, so these are the packed regions, and how the cells are actually processed. Based on that also you can take out 1 or 2 cell and then image it clearly. So in vitro is basically from the live cells. Ex vivo is from the dead animals, and in vivo, the third one is actually from the live mammalian family animals. In most cases, rats or mouse or opossum are used. So in this case, this first is a mouse image, and here they do some kind of on a full scale mouse, they make a small drill inside the brain, and they insert the microscopic probe, and they try to image the brain slice from that. So the in vivo imaging is the latest trend because you can keep the mouse alive and still you can actually image the live brain activities. I'm telling this is just a snapshot. 2D maximum intensity projection. So 2D MIP images are captured. So these are all 3D images, but you take a maximum intensity projection to generate a 2D image of the 3D volume. So I'll discuss both 2D analysis as well as 3D analysis. But in general, these are the three standard imaging modalities that are that people use: in vitro, ex vivo, and in vivo. In vivo being the most complex because of the complexity of the live brain structures and all. But in case of in vitro and ex vivo, you can do some post processing after extracting the brain tissue, and then the particular cell of interest can be actually processed and cleaned, so that the images are usually much cleaner in case of both in vitro and. Likewise, as I said, like these are the 3D imaging and 3D rendering of the neuronal cells. I'll come back to the same video later. Like this is the output of one of our methods. As you can see that you can clearly see the volumetric structure of the neurons, and these are the neuronal dendrites, and on the dendrites you have these small purple colored structures. These are called dendritic spines, and we attempt to actually assess the plasticity of these dendritic spines. As you can remember, the lecture topic of today is dendritic spine plasticity analysis. So these are the images of dendritic spines. These purple colors protrusions are dendritic spines. And what happens in real life, that these dendritic spines are not like this type of static image. So this is like one snapshot, and they really move like the tentacles of some like centipede. So okay, so they are they move like over time. So they try to search for the corresponding axon terminals. Okay, so the structure, position, everything changes for these structures, and they make a lot of biological meaning. And that is the core objective of the study. Can we quantitatively assess the structural changes in these dendritic spines? Okay, in 2D, in 3D, and over in 4D, that means over time. So that is the plasticity analysis. So what do you mean by synaptic plasticity analysis? That's just now, that's what I said. So it is well accepted that synaptic plasticity is essential for brain development and higher brain functions. So I was talking about degeneration of brain cells or brain activities. So what happens, that when a healthy adult tends to forget things, and when there is a neurodegeneration, that means the plasticity of the neuronal spines, the plasticity is affected. It is no longer plastic, so there is no major change, so they cannot change so frequently. So it is well accepted that synaptic plasticity is essential for brain development. Like for an early, in the teenage, probably the brain development and the synaptic plasticity is the highest. Okay, so new connections are made, and like the plastic changes can mean to new knowledge, new information gathering, everything is related to brain development. So synaptic plasticity is essential for brain development. So this is a well accepted biological hypothesis. So we depend on that. So we also know that changes in dendritic spine morphology are believed to be associated with synaptic plasticity. So this is the second hypothesis, that is dendritic spine morphology, that purple structures are dendritic spines, and the changes in the morphology means the shape of these dendritic spines are actually connected to synaptic plasticity. So that is also a well accepted biological hypothesis, and they are related to synaptic functions. Whether neuron connection, how it is made for exchange of neurotransmitter, that is based on this changes in spine morphology. I'll show how. Then this is hypothesis 2, and also the understanding of the relationship between changes in dendritic spine structure and synaptic functions remain inadequate. So this is the knowledge, in the underlying knowledge, and how these things happens and how this can be related. So this is still inadequate. So a lot of people are working essentially in this area. And effective solution to this problem is particularly difficult due to complex morphology of the dendritic. As you can see that the structures are complex. There is a so dense architecture, so it is difficult, and morphology is also complex, and the shapes are very difficult to map. And accurate quantitative analysis of dendritic spines are difficult and require extensive user intervention. So if you want to modify, if you want to quantitatively analyze, so there were a lot of user interventions required, and we significantly reduced this user intervention by developing several semi-automatic methods. So I'll show some of those methods, and we published several methods. And limited resolution in optical microscope. So even though we talk about like nanoscale up to nanoscale resolution, but it is still not enough, because the Z-plane resolution is not sufficient. So all these things are like key parameters of synaptic plasticity analysis, and we will come across these things whenever we discuss more. So some basics of what do you mean by the neurons. As you all know that there is a this is called the cell body or soma. So this is the main neuronal structure, the largest structure, and you have the nucleus inside, and then you have these dendrites. Okay, and you have the axon terminals. Okay, so you have, in general, a dendrite receives a signal. So you have the signals coming to the nucleus, and through axons, you send out the signal to the next neuron. So what happens if you look deeper? These dendrites also have these small structures, like these small protrusions. These are called spines. So these are the end of the dendrites, and at the end of the dendrites you have these dendritic spines. So they actually receive information from these axon terminals from a different neuron. So this is the story. Now, this axon terminal and this synaptic junction, they are responsible for exchange of neurotransmitter. Okay. If you see this in this particular image, we can image at this level. So this is the dendrite segment, and then you see that these are the spines. And these are the spines. Now you may ask, this is blank, this is dark, where are the axons? So this is a fluorescent microscopy. I'm just telling you that in this particular image, we are not interested to see the axon terminal. So it is not illuminated. The axons are not illuminated. That is why even if it is there, somehow it is not shown or illuminated in the microscopic image. So whatever we see here is just this segment, this part. from the dendrite. So this is the dendritic segment and this is the spine. Okay, so as you can see that this is an interesting story, that what would be the ideal shape of the dendritic spine so that it can effectively connect to, number one, to connect to the axon terminal and then exchange information. It is not simple. It is not straightforward. So for actual information exchange, it needs to have a binding, which unless there is a solid binding, no information is exchanged. Even if you have the dendrites, spines, information is not exchanged until there is a binding with the corresponding axon terminal. And there are different types of spines available, or the scientists have identified different types of spines. Here we will talk about at least 3 different types of spines. I'll come here, but again I'm just telling you, like one type is called a stubby spine. So this is called stubby spine. Then there is called a philopodia kind of spine. And then there is a mushroom type of spine. So as you can see that there are some spines. Say for these, this is a small stubby kind of a shape. So this is not actually connecting to the corresponding axon terminal. What happens? First, in the dendritic segment, these stubby spines are generated. Now, over time, these stubby spines get elongated in shape and take the shape of a philopodia. And when these philopodia or philopodiums are generated, they try to look for corresponding axon terminals. They just search for the axon terminals in the opposite side. And when they find somehow an axon terminal like this, then they change shape. So from here, philopodia, stubby to philopodia shape change takes place. Then philopodia to mushroom type, they change shape to mushroom. And when there is a mushroom type of spine, the axon terminal is also, they change shape like this and the information is exchanged. Okay, why mushroom shape? Because as you can see that this is physics, so that increases the surface area of exchange, information exchange. So a lot of chemicals, they actually exchange from axons to dendrites, these spines. And this region, you can see this is the neck of the spine, is also, they also play a huge role. So it is just like the transistor, they behave like a biological transistor. And the neck can be squeezed to limit the information exchange from here to, so how this information is exchanged? So when this is a mushroom spine, it gets information and it sends to the cell body. So this is the general story, and as you can see that dendritic spines change shape to connect to presynaptic terminals, that is the axons, and how they change shape, they are largely unknown, but people have different types of hypotheses, but this is the most acceptable hypothesis. And it is also assumed that neurodegenerative diseases like Alzheimer's, Parkinson's, like this type of diseases directly relate to the loss of synaptic junctions. So we call it demushroomization. So when the when the spines are not, there are not a lot of mushroom shaped spines like this, then we raise alarm. And on the other side, with age, people tend to lose a lot of mushroom spines, and only the mushroom spines contribute to the connectivity, information exchange. So if there is no mushroom spine, then there is no connectivity. And with neurodegenerative diseases, we lose mushroom spines, and that means loss of synaptic junctions. So that leads to memory loss. And how we can actually assess that quantitatively? How we can assess that? This is not a subjective story. We need to establish this hypothesis that neurodegenerative disease like this. So we are especially working on mouse models. So there are many different models, like we work mainly on stress and depression related behavioral models. They are experimented on the mouse, and that is a concept of you can check that whether you have a healthy mouse with happy living in a happy state, and then you can also create a group of stressed mouse living in a very stressful situation. How do you know the mouse can be stressed? So there are a lot of models where you can that can be used to as a stressor. There are different established stressor models. I'm just giving one example. Like big rats, they eat small mouse. Okay, so sometimes the mouse cage is kept inside the rat cage. So when the mouse sees the rat moving around trying to eat it, then the mouse gets depressed. So just imagine a human is placed inside a lion's cage over weeks, even if you are given food, so you will feel depressed, and you will get stressed, and a sustained stress leads to depression. So what happens? So when the mouse is depressed, so there are also experiments that validates whether a mouse is depressed or not. For example, I'm just giving you, so if a human being is depressed, then they stop eating. Okay, so there are some patterns, behavioral patterns to for depression. So same thing happens for mouse. Like normally mouse likes to drink sweet water. So if a mouse is depressed, they stop licking sweet water. Even if they're given sweet water, they do not lick it. So at that point, you can assess, okay, the mouse is depressed. And then they kill the mouse, extract the brain slice, extract the brain, process it in a specific medium. These are all in vitro experiments. Keep the neuronal cells alive and then image it. Likewise, you have a healthy mouse, kill the mouse, extract the brain, and then you have the control experiment, that is the healthy mouse situation. So then you have the stressed mouse, the depressed living cells, and then you can apply certain chemicals just like a antidepressant, and you can see whether the changes in the spines are taking place or not. So if the drug is right, then you will see that mushroomization is taking place. So what is the pattern of the spine structure? So this is the general model that people study, and we attempt to study. I was discussing more about general story of neuroimage analysis with respect to dendritic spine plasticity analysis. Now I'm going more into the quantitative analysis, what we do. So we have done one work that is called 2D span, that is for 2D segmentation, classification, and analysis of hippocampal dendritic spine plasticity. Okay, for the hippocampal region, mainly for the memory and other experiments that I have shown. So two papers, very interesting papers, especially the second paper where we published the method, and the method is also available for download. You can check 2dspan.org and you can download the method and use it, run with sample data. I'll show you how. And then we also worked with 3D span. That is another very interesting work where we worked on 3D spine plasticity analysis. Several interesting papers we published out of this, and very recently the tool, like this couple of weeks back in November, first week of November, we published the 3D span tool in this paper. This is a very interesting and very good journal, Neuroinformatics, and we could publish that. Before that, the theory was published in a Nature group paper, Scientific Reports, and another part of the theory was published in IEEE TFS. And another application of this was published by the German group in another Nature publication. So the method, you can see that in this paper, we first time shared this tool in the website 3dspan.org. It can also be downloaded, used. I'll share more about it later. So what is the concept in 2D span? Very simple. Now you think in a computer science perspective that a 2D MIP, that is the maximum intensity projection image of a dendritic spine, looks like this. And what we need to know, we need to extract, or first we need to segment these dendritic spines. How do we segment the spines? So the objective is to segment the spine and study individual spine plasticity. To do that, we adopted a method that is called digital flow. Okay, so you can click on one region, one point of the dendrite, the second point of the dendrite, and a hypothetical flow actually fills the inner region, and these spines can be clicked and they can be segmented. So you may not need, because this is a semi-automatic approach, and you do not need to actually segment all the spines at will, because the biologists need to select specific spines for longitudinal analysis. So this is our time varying data. So you analyze the data at time 0, so maybe after 10 minutes, after 40 minutes. So same cell is imaged over a specific time frame, and the same spine is segmented and analyzed over a period of time. So that's why you need to identify manually. So how do you do 2D flow analysis? So there we used convolutional kernels, complementary convolution kernels. So K11 and K12, if you see that this is in this direction and this is its complementary kernel in this direction. So if I apply from one point, so this kernel is going from say left to right, and this one goes from right to left, and we actually take the intersection of two. Likewise, this is for the up and down, this is for the 45-degree, and these are different. This is the 90-degree kernel and this is a 45-degree kernel. So these kernels are 90-degree kernels, and these are 45-degree kernels. So this angle is actually 45 degrees, and here it is 90 degrees. So you can see that how segmentation works. For example, if this is a dendritic segment and there is a spine, so you have a point P and a point Q, and if the kernel is spread over from P, you can see that partially this region is flooded. Likewise, if you have the complementary kernel, so you can see that this is the region, this orange region is filled by both the flows. So this is the intersection of both the kernels, intersection region, and this is the segmentation of the dendritic. Then you can click inside the spines and you can segment it. This is the general story of spine segmentation. You can see that there are different variations. There are a lot of experimental variability that you can. It also depends on. This is a binary data. So how you binarize the overall image, that is also an important aspect. So depending on the binarization, there are different adaptive binarization models that were used, and what are the flow thresholds? Depending on that, some regions are maybe over flooded or something. That can also be, that is also possibility. So to avoid confusions, so there are certain manual segmentation operations are also there. For example, here if a spine is touched like this, so there is no way you can actually separate this. Remember, the problem here is we are working on a 2D projection of a 3D data. So even if you apply the best binarization method, still because of the 3D architecture, there may be a spine which are overlapped in the 3D space. So if you take the projection, they will never be separated in the 2D space. So in such cases, you may need a manual segmentation operation so that these two spines can be separated. So this type of biological requirements means from the biologist sides, these are there. So our tool could actually segment these two through manual process, and these two are marked as a separate spine. Otherwise, all the other spines are separated like this. As you can see that on a sample region, if you mark two ends and then click on different spines, so they are separated like this. As I mentioned in the introduction region, that there are mainly 3 different types of spines. So you can see that these are called the stubby spines because these are real images, right? So these are the stubby spines. These are the philopodia or elongated spines, and these are the mushroom-shaped spines. Okay, so these are the mushroom-shaped spines, and for sure in these mushroom-shaped spines, so there are certain axon terminals are definitely there on the other side, which is not visible in this particular imaging modality. In this experiment, we also worked on a 4th type of spine. Some people work with spine head protrusions. They are kind of a mixture of mushrooms and philopodia. So these are called spine head protrusion. Sometimes what happens, you have a mushrooms, and then on the mushroom you have another elongation. So biologists still are unsure about why this phenomena takes place, but there are often some specific types of spines which are called spine head protrusion. So in this particular work, we were trying to mark also the spine head protrusion. But in sure, there are 3 types of spines in all the experiment, that is stubby, mushroom, and philopodia. Now, how we segment, like when we segment the spines, there are several key structural points that we need to define mathematically. Okay, so the first important point is the central base point. So central base point is basically I'm showing you like in this particular data, like if you have this segmentation from the dendrite and the spine, so this point is called the central base point of CBP of K is the central base point of the Kth spine. So this is one very important spine, because the first thing that you would require for a spine is the length of the spine. Okay, so for the length of the spine is estimated from the CBP, that is the central base point. Next is the central head point, that is the CHP. Now we call it basically the locally deepest point inside a spine. So if you have a spine shape, the locally deepest point is somewhere here. Okay, so we use fuzzy distance transform to estimate the locally deepest point in any segmented spine. Now, in some cases, there may be multiple locally deepest points, because a spine shape can be like, if it is a perfect round, then there may be only one locally deepest point. But if it is an elongated shape like this, there may be 1, 2, 3 multiple locally deepest points can be there. So what is the fitting the maximal ball? Okay, so probably some of you are aware of the skeletonization algorithms where we try to fit the maximal ball. So here, if there are multiple deepest points, then we try to average this head points. So there are multiple head points, and this is the average of these head points. So this is multiple locally deepest points P1, P2, P3, and we actually estimate the average of this as the central head point. And this is the FP, that is the farthest point. So the farthest point is defined as that this is the farthest from this particular central base point through central head point. So CBP, say FP is the farthest from CBP through CHP. So that we try to estimate, and these three define the most important structural points in the spine. And we try to assess many different parameters, not just the area of the spine. We try to assess the length of the spine, and what is the head width of the spine, what is the neck width of the spine. So all these things are assessed. You can see some of the examples, and we also try to classify the spines instantly. So yellow marks mushroom. Then you can see the smaller ones are basically, these are like stubby spines. These are like philopodia type spines. So all these types of spine classifications are done on a sample data. So this is the snapshot of the tool that we developed, and it is still available. If you wish, you can download it from the 2dspan.org, and you can just mark here and here on to end of the dendrite on which you want to work, and then you click, and all these segmented spines and the features will be estimated, and then you can also assess the plastic thickness. Now, to assess the efficiency of the developed method, we tried multi-user reproducibility. What do you mean by multi-user reproducibility? That the same tool on the same data set, we gave it to 3 independent users, and they tried to mark the spines. Okay, so since we are working with the Polish lab of Nensky Institute of Experimental Biology, so we had the biologists working there, and they tried to segment the same data, and we tried to assess the final segmentation result. And as you can see that if you talk about the area at time 0, more or less they are same. In some cases, as you can see that in some spines there are like lot of variability, huge variability. Okay, more than 0.5 microns, so micron square in terms of area. So this type of variability was noticed, but in general we saw like as you can see that in most cases the variability is accepted. Now, as you remember that the topic of today's lecture was plasticity analysis. So what do you mean by this plasticity analysis? Plasticity says that how the morphological changes in the dendritic spines can be analyzed. Okay, how the morphological changes can be quantitatively assessed and analyzed. That means, suppose we have the same cell, as you know that in vitro images, you can keep, although the animal is sacrificed, but the brain cell can be kept alive in a medium. Okay, so in that medium, if you process the with certain chemicals, the cell will remain alive for a certain amount of time. And after treating with a specific chemical of interest, say an antidepressant, you want to assess whether there is any changes, whether there is any change in the morphological structure of the cell. That means of the dendritic spine. For example, if we talk about baseline, that means the time 0. So this is the baseline, that is in time 0, we tried to assess what are the composition. So we saw that there are 31% stubby spines, 32% philopodia, and around 27% mushrooms. 27% mushrooms. And after 10 minutes, we assessed the same cell. All the spines that were marked in time 0 were again marked at time 1. So we, in the microscope, what happens? The experimental setup is the slide is placed in the microscope, and first we image it at a given time 0, and then we image it after 10 minutes, then again after 40 minutes. Okay, so after 10 minutes, when we are assessed, the same spines were marked again. We saw that there's a slight change in stubby spines, so some more stubby spines could be marked. Philopodia is also 32 to 33, mushroom spine population slightly decreased. So you can see that what are the changes, like some of the mushrooms, 12% mushrooms remained as mushrooms, some of the 21% stubby spine remained in the same stubby state. Some of the, this is important, how much of the percentage of the stubby goes to philopodia. So we could see that the 5% of the stubby spines go to philopodia, and from philopodia to mushroom, around 6% of the philopodia goes from philopodia state to the mushroom state. So these things was noticed. So this is T0 to T10, but we could not reach a reasonable hypothesis from this particular T10, T0, T10 analysis. Then we extended it for 40 minutes. So T0, we imaged it, timestamp 1, timestamp 2, T10, then timestamp 3, T40. So we analyzed it at T40. And then we tried to see that, okay, in T0, we had 48%. In another experiment, 48.7% stubby spine. And in T40, if you skip T10 state, and then you see that the mushroom spine population was 17.9, and now the mushroom spine population is 35.9. And you see the Philopodia was 23%. The Philopodia is increasing to 30%. And then the mushroom spine is increasing. So you can see that if you keep it in the longer, for a longer period, up to 40 minutes, then there is a change from the, see the stubby population reduces by almost 15%. Okay, and the mushroom population increases from 17% to it almost doubles, and the Philopodia increases and then decreases because some of them goes to mushroom. Okay, so this is an interesting observation that from T0 to T40 we could actually see a noticeable change in the mushroom spine population, and probably the effect of the chemical takes a bit longer time to settle down and make an effective change in the spine morphology. Okay, so if you relate it back to the drug design and all, so you will see that some of the drugs you take and it takes some time to make the effect, right? So some drugs take a longer time to take the effect. So if you consume a drug, then to make that effect into the certain to make some biological changes, it takes time. So that way you can make an inference that there is it takes more time to change the morphological structure of the dendritic. So we attempted to compare the results with some benchmarks, and I'm just skipping this and summarizing. Like digital flows successfully segments the dendritic spines from the dendroids. It's a fast, user-friendly, and accurate method to with respect to available state-of-the-art. Multi-user reproducibility score was high, and formal morphological definitions of spine compartments were provided, and effective analysis of spine plasticity and possible extension to 3D flows or in general 3D segmentation. So 2dspine.org, I'm not going to the site, so if you wish you can just visit the site. It's not a well-designed one, but you can still download the software and have some test data, some testing data is also, the Windows executable is provided. Now if we move to 3D, the same concept is valid, but we are working on a quantitative 3D morphometric analysis. So here, what is the target? 3D segmentation of hippocampal dendritic spines using a novel multiscale opening. So I mentioned there was a IEEE transaction paper, IEEE TFS paper. So that details the 3D segmentation algorithm developed by us, and then we used the definitions, or we extended the definitions already given in 2D to the 3D domain, and the developed model is to be validated using confocal light microscopy image of dendritic spines for dissociated hippocampal cultures for qualitative and quantitative analysis of spine morphological changes before and after chemically induced CLTP. That is like in a simplistic term, like when you apply a chemical and what is the effect on application of the chemical. Okay, so reproducibility analysis and accuracy analysis. So why 3D? Why 3D? The simple reason is shown here. As you can see that in 2D, this is the 2D MIP. 2D MIP is maximum intensity projection. So if you see this, the maximum intensity in Z direction in Z is projected. So this is the MIP. But if you see that what is here, so these are the completely different structures. So you see that you see much more in 3D because of this volumetric rendering. So it is always expected that you, if possible, you work directly in the 3D. In microscopic images, you anyway are getting the 3D images, but it is extremely challenging to render the 3D data as well as work with the 3D data. Okay, so people mostly work with 2D MIP images. We proposed one of the first methods that could analyze individual spine plasticity in 3D. One of the first methods in this domain. So I'll show more about this. So I'm not going in details about the 3D segmentation algorithm. The 3D segmentation algorithm is not related, not really connected to dendritic spine plasticity analysis. It is just a 3D segmentation in the shared intensity space. So we work in the model that there is a in the intensity space, the dendritic core is of high intensity and the spines are of lower intensity. So if we have a thick core, so we can identify some region of pure dendrite region, and rest this is a shared intensity space where both those intensities can be both part of the dendrites as well as from the spines. So we are not sure, but if we have a shear shot high intensity region for the dendrites, so we can use them as the dendrite seeds. But in some cases, in some structures, pure dendrite region is hard to find. And in those cases, we do not have a pure dendrite region. The full region is considered as a shared intensity space between spine and dendrite. In that case, we need to give some seeds to dendrites and some seeds for spines. So some sample of 3D segmentation result of the same data that we showed here and here. So this is the general theory, and the basic theory is revolves around the fuzzy distance transform or FDT in the intensity space shared by the fuzzy objects, in this case the spine and the dendrite. And the major challenge is how to separate the spine from dendrites, which are fused at various unknown locations and scales. As you can see that some connections are like this, where the segment scale is really large, and in some cases you really have thin regions and the scale is really low. So finer scales and the larger scale are possible. So that is a challenge at times, and that is why we need actually a multi-scale operator. That multi-scale algorithm we proposed in 2015 and 16, And the first step is segment the combined region O spine union O dendrite from the background, because the background we first need to separate, and then separation of O spine from the object spine from the object dendrite of the MSO algorithm. So it actually works it actually works in the fuzzy connectivity space, and it iteratively opens up two objects. If you need to go deeper, you can read the papers. I'm not going much into the detail. But in general, if you see that the structures like base of the spine, central base point, central head point, and the farthest point, they all hold good both in case of 2D as well as 3D. And we actually can segment the structure. Suppose this is the segmented spine, and if you can see that if you remove the transparency a bit, play with the transparency a bit. So this is the way how the spine is actually segmented in 3D. And this region is the central base point, and this is the farthest point. So this is the segmentation result that we generate, and this is how the segmentation is actually affected in the 3D object. If we go, if we analyze further, you can see that you remember that I was talking about the maximal ball that can be fit inside the head in case of a mushroom spine. So this is a simulation of the head and the neck region. So this is the central base point. So this is the head neck junction, and this is the core or the skeleton of the neck, and this is the maximal ball that can be fitted inside the mushroom head. So we have many interesting structural points here. You have the central head point, you have the farthest point, you have the head neck junction, you have the neck region, you have the central base point. This is just a representative image of merging both of them. If you extend this for many different types of spines, we call it, as I said, like we worked with stubby, filopodia, and mushroom. So we also try to define another kind of spine which is neither stubby nor filopodia, and we call it a thin spine. But as I also mentioned, like stubby to filopodia and filopodia to mushroom, this is a very standard logic, and that we try to follow all the time. Okay. Sample results, as you can see that from 2D MIP, and this is a small region of interest, and this is the final segmentation result. And as you can see, like all the spines, spine number 5 is probably a mushroom spine. As you can see that spine number 5, what is the volume, what is the length, what is the head width, what is the neck length, what is the minimum neck width, what is the average neck width, and what is the type of the spine. So this average neck width of half a micron is or 520 nanometers is really important, because these spines, they automatically tweak this neck width, and depending on the neck width, even if you have an interconnection, you can control the information exchange. So sometimes the average neck width increases, sometimes the average neck width decreases. So it all depends that what is your head width, and then what is the neck region, and you can see that some head width is large. Here, more or less, like this is a slightly smaller spine, smaller mushroom. So these are like, these two are more or less of the same scale, and the neck widths, average neck widths are also same. But as you can see here, especially here, you can see that this neck width can be tweaked, or biologically the mammalian body, the spine neck width is actually adjusted so as to control the exchange of information, just like a transistor. If they, like a bottleneck, they can control the amount of information that needs to be. The weights can be, we all are aware about the artificial neural networks. So the network weights are adjusted with the basis of this neck width. Especially the minimum neck width is also important, that there is one constrictor. So there is one constant region. See, 1.194, this much is the minimum. That means this is the information flow is regulated by this. So this spine, even though it is slightly having larger head width, is still having less minimum neck width. So information exchange is less in this spine number 4. So this is more or less the story, and these are the segmented spines of different categories. You can see that stubby spines, filopodia, spine head protrusions. As you can see, that some examples of spine head protrusions that I was discussing. 2D analysis. For accuracy analysis also, we try to mark several spines. For segmentation accuracy analysis, we try to mark ground truth points in each spines and its surroundings in the ideal situation, and then attempted to assess the accuracy. So in some cases, you can see that in 3D, you can see there are often errors in the volumetric measurement. Okay, because sometimes the volume analysis, there are certain problems, but in general the length is more or less okay, except some 1 or 2 errors. So this is the ground truth, and this is the conformity with the ground truth that we show. Now, there are certain benchmark tools available, but as I mentioned before, that there is ours was one of the, or still like one of the prominent tools still available here, which can accurately map the 3D structures. And see, Imaris is a very popular bitplane from the bitplane group. So this is a very expensive commercial software, but still they fail to model every spine. So because they have some standard models that fits a spine. So in some cases, if you have an arbitrary shape, then the spine does not fit that particular model, and Imaris fails. So that is a qualitative comparison with respect to a state-of-the-art tool. And if you talk about reproducibility in 3D, we gave the same segment to 3 different users, and we tried to assess the performance of the 3 users. And as you can see, more or less, you have the conformity, except in some cases, especially this is the length. See, there are some errors here. Also some problems in case of head width. So in general, you can see that also in the volumes, there are certain issues, but in general, in many cases, you can see that there are conformity and multi-user reproducibility holds good in general. And as you can see that plasticity analysis in the baseline after 10 minutes, after 40 minutes, you have you can see that some of the spines they are developing in changing in shapes. See, there is a spine head protrusion that is getting generated. So you can see that this particular spine is in getting bigger and bigger. See, spine number 4 is getting bigger and bigger. So all these things are significant, computationally significant. And average changes in individual spine morphology, that also I average changes in individual spine morphology, as you can see that in baseline after 10 minutes and after 40 minutes, as I discussed even in case of 2D, that after 10 minutes there is also some kind of misinformation. So you can see that the spine volumes in general they increases after 40 minutes. Likewise, the spine length also increases and the head width also increases. In general, overall, we try to assess like what is the changes in individual spine morphology. So in all spines after application of these chemicals, we changed the spine heads length and volumes were changed. Some more experiments are very specific, like one single spine if I identify, and this is one of very recent Nature publication from our group. We could see very specifically that after application of a certain drug, so there is a change in spine morphology, and the same spine is before and after the drug stimulation, it is changing. So as you understand already that our expertise is not in design of the drugs, neither in design of the mouse model experiments, but our objective was to quantitatively analyze and give or generate a tool for the biology so that you can effectively see in 2D as well as in 3D, yes, there is a change in shape after stimulation. So this is this was the core responsibility, and this is the part of the work that we are actually doing. So this is also data, in vivo data. Most of the data that we have shown here are in vitro data, but here these are some more complex data with Hannover Medical School in Germany, and here also we could assess the segment the spines, and this is a final result from this in vivo, that is the live cell imaging data that I showed in the first slide, and result of our multi-scale object MSO, that multi-scale segmentation, and our 3D span tool were used in combination, and we generated this video rendition of the data that is actually from a live mouse experiment. Summarizing with 3D span, so 3D span algorithm successfully segments and extracts key morphological points in its segmented spine. So accurate estimation of length, head width, neck length, neck width features. So detailed analysis of spine plasticity, accuracy, and reproducibility analysis is presented. And if you visit 3dspan.org, you will see that the latest version of the tool is available. The tutorials, the video tutorials, all the help files, sample data, code is also available. We developed this using the Qt IDE, and it's basically a C++ based tool. Also, we used Python Qt binding also used. So the complete software and the Windows executable is available. The code is also available, the total project code, and that is that can be directly because Qt is platform independent. Even in Linux environment, it can be run. So we are means especially I'm very happy to share that when we do research, so one of the major objective of research is like whether that creates an impact or not, whether people are noticing your research or not. So we are very happy to say ki Dr. Catherine Willig, she mailed me directly for inquiring about the 3D span tool when we first published our paper in scientific reports. That's a Nature group journal, and Catherine Willig mailed me and wanted to know more about the tool. And Catherine Willig is the first author from the paper of Dr. Stephen Hull, who got 2014 Nobel Prize in chemistry for invention of the state microscopy. So the microscopic data on which we are applying our method, that is the 2-photon and multi-photon microscopy. So Stephen Hull is the pioneer for that work. And in Stephen Hull's group, Catherine Willig was the first author of that paper. So she is from Göttingen. So she mailed me and to our collaborators and wanted to use the tool. What are the benefits, etc. So we are very happy that people are working with our tool. And Tahare is a postdoc from the group of Erwin Nehar. So they also mailed me and my collaborator in Poland. So they are also from the group of Erwin Nehar. So Erwin Nehar is a very old Nobel laureate, and so they are also working in this particular domain, and they're also from the Göttingen incidentally. So probably we have some exposure in Göttingen, and 2 Nobel laureate groups, they contacted us. Many people are actually using this tool. We are getting regular emails, and we address queries from many different groups. I'm also very happy to share that Daniel Rubin is a very famous Stanford University professor. He also mailed me and wanted to know more about this particular tool and how to use, like what utility and advantage. Maybe there for the comparisons and all. So I'm not claiming big, but I can at least showcase. So they are like recommendations for our work, our effort, and that is what matters in research, that whatever you do, if people recognize and notices your work, so that gives a huge satisfaction. So with this, I sincerely acknowledge my international collaborators, especially from Nenski Institute of Experimental Biology. Dr. Jakub Lodarczyk is my long-term collaborator, and they are doing all the mouse model experiments there at their end. So they are pure biologists, so with limited computational knowledge. So we have a very good complementary skill set. So we develop tools for, we try to solve a lot of computational problems that they face on real life, and they use some of the tools that we develop. And Andre Zug and Evgeny Ponimaskin are from Hanover Medical School. They are also biologists. Andre Zug is actually a physicist and mainly expert in the microscopes. and microscope handling. So during my DAD fellowship, I visited Germany and got an opportunity to work with them, and we have several excellent publications together. Professor Poonam Saha, with Professor Poonam Saha, I developed that multi-scale 3D segmentation algorithm did during my first postdoc in 2010-11. And he is my long-term, close to 15 years collaboration we have together, and we still collaborate actively and publish not just in this neuroimaging informatics, but also in other domains. And Professor Dariusz Plewczynski is also from University Warsaw. I did my second postdoc with him, and he is also associated with this project. And he is a true signature of interdisciplinary research. He is having background in physical chemistry, then both physics, chemistry, then biology and computer science. So he is a excellent mix, and I'm collaborating with him for close to 10-12 years now. So he's also my long-standing collaborator. And my former PhD students like Dr. Shatudal Shaha and worked in this particular domain and did a postdoc in University of Warsaw. So was Dr. Molla. So he also visited University of Warsaw and for his postdoc. And Dr. Pranati Dokshit also worked in this particular domain briefly, and she finished her postdoc PhD a couple of years back, and she's with the IS right now. And ongoing in this particular domain, I'm currently working with Mr. Nirmal Das and Shobhik Paul, and some of the tools are developed by my past graduate student, Mr. Aditya Rala. So I sincerely acknowledge all the contributions from my students and collaborators. And thanks for your patient hearing. In case you have any query, you can always mail to me, and thank you very much.
Thank you, Professor Basu, for this wonderful lecture. It was really informative. Okay, so I would like to know from the participants whether there is any question from you. You can also write in the chat box. Sir, since there is no question from the audience, so so with your permission, may we end the session?
Yes, sure, why not?
Okay. So thank you very much, sir. So we have a small token of appreciation for this wonderful presentation. We would like to have you in our next FGB sessions also. Thank you very much, sir.
Okay, thank you very much. Thank you. Okay. So I think I can leave now. So thank you. Thank you
Yes,
for
yes.
invitation. Yes. Okay. Thank you.